Hello, my name is Samantha Valencia. I'm a biology major at Oglethorpe University, and today we will be reviewing a study that I found called In-Lake Algal Bloom Removal and Submerged Vegetation Restoration Using Modified Local Soils. So this study was published by Ecological Engineering in 2011, and the authors are listed below. The reason why I chose this study was because I was interested in global climate change. And throughout my research, I found that global climate change causes algal blooms. And there was a growing concern in the number of algal blooms that are formed. And now I found this article that actually got rid of algal blooms, so it was pretty neat. And so before I go off in our presentation, I'll go through an outline that basically highlights how this presentation will go about. So first we'll go over why are harmful algal blooms dangerous for the environment? How are harmful algal blooms formed? What are ways algal blooms are treated? How does chitosan cause flocculation of algae? Experiment introductions, experiment methods, experiment results, some conclusions, and takeaway ideas. So we'll begin with why are harmful algal blooms dangerous for the environment? And they're dangerous for the environment because they release toxins. In this study in particular, they measure the amount of microcysteines, which is a type of toxin. These toxins can kill organisms and humans. They also promote some acute problems, which is hemodynamic shock, which is basically the decrease in blood flow within an organism, which causes some sort of vital organ failure. Another acute problem would be heart failure, and lastly would be death. Some chronic problems would be liver inflammation, and it would also promote tumors to be more prevalent in people who are exposed to these toxins. And lastly, we'll go, these harmful algal blooms cause a decrease in oxygen levels, and this decrease in oxygen levels lower the amount of vegetation, so it doesn't allow these plants to survive in the water. And organisms also die because they don't have sufficient amounts of oxygen, and with these decreased levels of oxygen, cyanobacteria, which is a type of algae, it thrives in that type of environment. So now we'll go over how our harmful algal blooms form. They're formed through an accumulation of different types of algae. In this particular study, they mentioned cyanobacteria is the type of algae that is accumulated. And you might ask yourself, well, why are these cyanobacteria being accumulated? And this is because they are put in environmental conditions in which they thrive in. So cyanobacteria thrives in high temperatures, like above 25 degrees Celsius. They thrive in high light exposure for long durations. These algae thrive in stable water, so They like slow-moving water as compared to fast-moving water like rivers. And harmful algal blooms are also caused by an excess of nutrients, which is also called eutrophication, and this occurs through runoff. And runoff is basically when, let's say, agricultural industries use fertilizers that cause the soil to have excess nutrients, and this soil runs off into a body of water, and the excess nutrients are now within the body of water. Just to recapitulate what I previously mentioned, I found this flowchart that might help us better understand how algal blooms form. So this flow chart actually includes global climate change. We see this on the left-hand side of the image where they show agricultural, industrial, residential, and urban sources creating an increase in NH3, NO, NO2, and CO2, which cause a greenhouse effect in the atmosphere and ultimately causes the water to become warmer. As I previously mentioned, algae likes warm temperatures, and from this image we see sunlight hitting the algae, and algae likes long durations of high intensity light exposure. From this image we can also see runoff on the left hand side, where there's an arrow with nitrogen and phosphorus within it. So these agricultural areas use, for example, fertilizer, which causes an increase in the amount of nutrients on the soil, and the soil ultimately runs off into the bodies of water and increase the amount of nutrients within the body of water, leading to the increase in the amount of algae in the body of water. Another idea that I didn't previously mention was that algae, when it dies, it sinks down to the bottom of the bodies of water, and decomposers decompose the dead algae and release more nutrients into the water as well. So as you can see, it's very complex, the amount of factors that play into the growth of algae, which makes it very hard to try and get rid of it. In particular, cyanobacteria, it survives in various forms of temperature and light exposure, which also makes it very difficult to get rid of cyanobacteria, which is a type of algae, a specific type of algae. So now we'll go over what are ways algal blooms are treated. First, they are treated the first way. They're treated by. So now we'll go over what are ways algal blooms are treated. They are treated by algicides. Algicides is one way algal blooms are treated, and algicides are basically predatory interactions between bacteria and algae, but this can have an adverse effect because the lysing of the algae caused by the bacteria ultimately releases toxins, and that could harm organisms in the bodies of water, as I previously mentioned. And a second way algal blooms are treated are through filter feeding fish, mollusks, and coral. However, this form of treating algal blooms doesn't work on harmful algal blooms because if they ingest these harmful algal blooms, they can ultimately die, and it's harmful towards these organisms. Another form algal blooms can be treated is through chemical flocculation. So flocculation is basically the clumping together of algae, and once it clumps together, it sinks down to the bottom of the body of water. And they do this through chemicals, which isn't natural, and it could have adverse effect on other organisms in the bodies of water. It is found that in chemical flocculation, it's more likely to be successful in the laboratory than it is in real life, and it could be very expensive to do the previous 3 treatments that I mentioned. So lastly, modified local soil induced ecological restoration is another way that algal blooms can be treated, and it's the way that it is treated in the study that I found. And if we simplify and look at what is in its title, we see that it's modified local soils. So these modified local soils have some sort of polymer that causes the flocculation of the algae. Along with that modified local soil, they add macrophyte seeds, which they expect to germinate once the algae sinks down to the bottom of the body of water. And this ultimately would cause an ecological restoration. So overall, it would help get rid of algae, and it would help restore the ecology of the water by increasing vegetation through the macrophyte seeds. So now we'll go over how does chitosan cause flocculation of algae. In the study, chitosan is the modified local soil. So it's what's included in the modified local soil. In the study, they use chitosan, and chitosan is a biopolymer which is made out of seashells, and it comes from seashells. And these biopolymers have long polymers that wrap around the cyanobacteria and cause them to clump or coagulate and ultimately sink down. So, as you can see from the image, I added an image that that goes through chemical flocculation. It's not the same as the chitosan flocculation that was used in the study, but it's the same idea where you see the raw water flocculating and ultimately sinking down to the bottom of the body of water. Finally, we have made it to the experiment, and this experiment was conducted in Taihu Lake in China, specifically Liaoyang Bay. And this city suffered through malodors, and they saw fishes floating up above the shallow bay. And they wanted to see if they could use this modified local soil-induced ecological restoration technology to help treat these harmful algal blooms. So their goal or hypothesis was to study the efficiency of this technology in treating the water, and they wanted to measure its ecological effects in the restoration of the macrophytes in the water and the quality of the water resulting from this treatment of modified local soil-induced ecological restoration. Here's an image that shows the bay. So they said that the accumulation of algae was caused by wind blowing the algae into the bay. And so before conducting the experiment, they closed off the mouth of the bay in order to prevent more algae to flow into the bay. And they had 3 sites within the bay where they measured the concentrations of various things in order to see the short-term and the long-term effects of the treatment. And control number 1 and control number 2 are other sites in which they measured the concentrations of various things to see the effect, to measure the effects outside of the bay. And they're the controls of the experiment. So the methods. Basically, they had 2 containers where they mixed the chitosan solution with the macrophyte seeds in some lake water. And with this technology, it merged all 3 of those into one solution in which they were able to spray on top of the water. And you can see from this image that previously it was all covered with algae, but once you spray it, you could see the noticeable difference in the color of the water. And so they did the spraying twice in order to prevent the algae to resuspend. I added this image as well, which is 1 day after the treatment, and you can see the difference in the color of the water. And here are images before using the treatment and images after using the treatment. And so here is the data, and in this data it's the long-term effects that are shown, and their long-term effects were only measured within the time period of 4 months. So it's not that long, and the only reason why they only measured it for 4 months was because the government decided to turn the bay into a water park. And okay, so from this data, they measured the various amounts of nutrients in the water. So they measured the concentration of these nutrients in the various sites. So site 1, site 2, and site 3 are within the bay. Control number 1, control number 2 are outside of the bay. And in all of these charts, within the bay, we see that the concentration of these nutrients start off high and decrease drastically. Outside of the bay, the concentration of these nutrients start off low and decrease throughout time as well. Through statistical analyses, they found that it was significantly lower. They found that within the bay, they had significantly lower amounts of nutrients than outside of the bay. So this was good for them in showing that they were able to maintain the concentration of minerals low throughout the time period of 4 months. They also measure the amount of, the concentration of the toxins found in these various locations. So microcystine RR is one type of toxin, and they measured the concentration of that toxin in site 1, site 2, and site 3, which is within the bay. And control number 1 and control 2 is outside of the bay. And this was measured within 4 months as well. It is seen that throughout the time period for both microcystine RR and microcystine LR, the concentrations increased, but overall they were lower within the bay than the amount of concentration that there was outside of the bay. And they found this because during the winter time, which is around December, most of this algae had already died off, and when it dies off, it releases these toxins. But since it is overall lower within the bay, they find that this treatment is still helpful and it's not overall harmful in the release of toxins. They also measured the biodiversity index inside and outside of the bay. So the biodiversity index was measured through the Shannon-Wiener equation, and that's not particularly important, but it's just a reference on how they measured that index. And they measured the biodiversity inside and outside of the bay. So the biodiversity of phytoplankton, zooplankton, zoobenthos were measured, And overall, during the time period of 4 months, they found that within the bay, the biodiversity increased for these various organisms. And this is mostly due to the decrease in the amount of algae in the water. The decrease in the amount of algae in the water basically gave rise to these organisms being able to come back to their biological environments. And it also proves that chitosan was a safe flocculation method, and it's safe for the organisms within the bay, and it's not harmful towards them. And now we've learned that harmful algal blooms are increasing because of global climate change, and these harmful algal blooms cause acute and chronic problems. These harmful algal blooms could effectively be treated by MLS-IER technology, which is modified local soil induced ecological restoration technology from the study previously mentioned. And they not only help get rid of these harmful algal blooms, but they also help other biological organisms return to their environment. Thank you for listening.